Hey guys, just wanted to share a couple helpful hints for the genetics counselor project. So first we want to start with some useful vocabulary. First thing is homozygous. If a characteristic is homozygous, that means that the organism has two of the same allele. So if they have two of the dominant allele, we're going to call them homozygous dominant. If they have two of the recessive allele, we're going to call them homozygous recessive. So let's look at an example. Here we see two capital D's. That's going to symbolize homozygous dominant because I have two of the dominant alleles. Here I see homozygous recessive because I have two of the recessive alleles, denoted by the lowercase a's. I could also have an organism that's heterozygous. That means that the organism has one of each allele. So they have one dominant and one recessive allele. So let's look at an example. Here you see we have one capital T and one lowercase t. So that denotes that it is also heterozygous. So we also want to look at the difference between a dominant allele and a recessive allele. If there is a dominant allele present, then that trait is going to show up. And then a recessive allele traditionally gets masked when a dominant allele is present. So if there is no dominant allele, then we will see the recessive trait. So here we see two capital S's. So that means we're going to see the dominant trait. Here I have a capital S and a lowercase s, our heterozygote, and that's also going to show the dominant trait because it has one of those dominant alleles. And then here we see two lowercase s's. It's going to show the recessive trait because it's homozygous recessive and it only has the recessive allele. So traditionally, when we were trying to determine which letter to use for our symbol, we see what's dominant and we use the symbol that denotes the dominant trait. So if we're looking at tall being dominant, the symbol is going to be a capital T for tall, and then we'd use a lowercase t for the recessive allele for short. But for this activity, I want you guys to always use the first letter of the disorder. So capital letter will be denoting a dominant trait, and a lowercase letter will denote the recessive. And so it doesn't matter if the disease is dominant or recessive, just use the same letter. So for example, arthritis. The letters I would use would be capital A and lowercase a. Capital A would denote dominant and lowercase a would denote recessive. And I would have to figure out whether arthritis is dominant or recessive to determine whether an organism would have arthritis with either of these alleles. Another example would be diabetes. The two letters I would use would be a capital D or a lowercase d. Capital D denoting the dominant and lowercase d denoting the recessive. And again, I would have to figure out whether diabetes is dominant or recessive to be able to determine what the organism is. So here let's do an example. So if we look at arthritis and we'll assume dominant, I honestly do not know if it is or dominant or recessive. I was just using different diseases as my example. So here we're going to cross a homozygous dominant with a homozygous recessive. So the first step is, what symbol am I going to use? Since I'm talking about arthritis, I'm going to either use a capital A or a lowercase a. The second step, I'm going to write the genotypes for the parents. So since I have one parent that's homozygous dominant, I'll have two capital A's. And since the other parent is homozygous recessive, I'm going to have two lowercase a's. The third step is going to be placing those parents on the Punnett square. So I'll draw my Punnett square, and then I'm then going to add the parent. So homozygous dominant will get added once, and then homozygous recessive will be added on the other side. Traditionally, the male will go on the top of the Punnett square, but it truthfully does not matter. The next step is going to be to cross the parents by completing our Punnett square. We're first going to pull these capital A's across. So we'll pull those across to fill in those squares, and then we'll pull the other capital A's across to fill in those squares. We'll then pull the lowercase a down to fill in those squares, and then we'll pull the other lowercase a down to fill in those squares. So the next step we're going to do is step 5. This is where we're going to determine the phenotypes. So since I know arthritis is dominant, any one of these offspring that have a dominant allele are going to have arthritis. So I can say that 0% of them are normal, 0% of them are a carrier, and 100% are affected because every single one of those offspring has that capital A denoting that it has arthritis. So here let's look at example 2. Example 2 looks at diabetes, and we're going to assume that it's a recessive trait. And we're going to cross two heterozygotes. So heterozygote crossed with heterozygote. Again, step 1 is determining what symbol you're going to use. So since we're looking at diabetes, we're going to either use a capital D or a lowercase d. Then we'll go to step 2. Step 2 is writing the genotypes for the parents. Since they're both heterozygous, I'm going to write out a capital D and a lowercase d for the first parent, and a capital D and a lowercase d for the second parent. Then we move into step 3. You're going to then place the parents on the Punnett square. So draw your Punnett square, put one parent on the left, one parent on the top. Again, traditionally, father goes on the top of the Punnett square, but it does not matter. You're going to pull the capital D from the left across. Step 4, cross the parents by completing the Punnett square. So we'll pull the capital D across, and then we'll pull the lowercase d across. We'll pull the capital D down, and then the lowercase d down. And so step 5 involves determining the phenotypes. Since diabetes is recessive, the only organism on here that would show the diabetes would be the homozygous recessive at the bottom. The one that is heterozygous would be considered a carrier, and if you're homozygous dominant, you're going to be normal. So I have 1/4 or 25% is normal. I have 50% is a carrier, so heterozygous, and then 25% is affected or homozygous recessive. So I hope that was helpful.